Retroviruses are a group of viruses found in the family Retroviridae, which possess the RNA as genetic material, and they have reverse transcriptase to convert the RNA into DNA to be integrated into the host. There are two genus here, the genus Lentivirus and genus Delta Retrovirus. The genus Lentivirus has the HIV-1 and HIV-2, and the Delta virus genus has HTLV-1, which is the human T cell lymphostrophic virus. Coming to the structure of HIV, so we studied two things in the structure. Of First of all, it is spherical in shape, and it is 80 to 110 nanometer in size. In the structure, we study the envelope and the nucleocapsid. The envelope has a lipid part and a protein part. The part forms the lipid part is formed from the host cell membrane. So this is the lipid part of the cell membrane, and in the protein part we have two glycoproteins, glycoprotein 120 and glycoprotein 41. The glycoprotein 120 is projected as knob-like spikes on the surface, while glycoprotein 41 form a anchoring transmembrane pedicles. So these are the transmembrane pedicles formed by the glycoprotein 41. This was the lipid layer, and here we have the glycoprotein 120 forming the knob-like structure on the surface. Glycoprotein 120. So these three things together form the envelope. Talking about the nucleocapsid, we have a core protein which encloses the RNA, which is actually two copies of positive sense linear RNA, and we have viral enzymes like the transcriptase, integrase, and protease. If we draw this is the core. It has two positive sense linear RNA and the nuclear enzymes. What we missed here was drawing the matrix beneath the lipid layer. Just completing that. Now labeling the nucleic acid. This is the core. These are the RNA, and this is the nuclear enzymes. Talking about the genes and antigens, so we have structural genes which form the structure of the HIV virus. There are three structural genes: the GAG gene, Paul gene, and ENV gene. The GAG gene has uh, it is expressed as a precursor protein P55, which is cleaved into three proteins: P18, P24, and P15. The P18 forms the shell, while the P24 and 15 form the core. The Paul gene is expressed in the viral enzymes and is cleaved into three proteins: P31, P51, and P66. The 31 codes for integrase, 51 codes for reverse transcriptase, and 66 codes for protease. The ENV gene has P160, which is cleaved into P120 and P41. The P120 codes for it's actually glycoprotein 160, which is cleaved into glycoprotein 120 and glycoprotein 41. And glycoprotein 120 was the knob-like structure, while this was the transmembrane protein. Talking about non-structural genes, we have a regulatory genes which are essential and three accessory regulatory genes. So in the essential regulatory genes, we have TAT, NEF, NEF, and REV. TAT is transcriptional transactivator. NEF is negative factor. And REV is the regulator of virion expression. And in accessory genes also we have three genes: the VIR, VPR, sorry, it's the VIF, VPR, and VPU. VIF is viral infectivity factor, viral protein R, and viral protein U.